Good morning, everyone. Today I'm going to talk about the elevated immune response in octopus rubescens under ocean acidification and warming conditions. As we know, human activities has released tons of carbon dioxide into the atmosphere, and it has rapidly increased the concentration of the carbon dioxide in recent years. And there are researches on the impact of ocean warming and acidification on calcifying marine organisms. For example, the corals and the bivalves. But we know little about how climate change will do to those invertebrates, such as cephalopods. The aim of this study is to understand the impact of ocean acidification and ocean warming on the invertebrates like octopus. And what is the importance of the octopus in the ecosystem? Well, octopus are a group of marine mollusks that can be easily found in many depths and in most marine habitats. And in the food chain, they are not only the predators, but also the important prey for many other species, like seals, sharks, whales, seabirds, and other big fishes as well. And they are an indispensable link between trophic labor in many marine ecosystems. And why the scientists chose Octopus rubescens to do the study? Because it is one of the most common octopus species of North America West Coast. And where the octopus were collected. The octopus were collected by scuba from Driftwood Park, and then they were transported to Rosario Beach Marine Laboratory. And in the experiment, only octopus with a mass of 150 grams or more were used. And because they didn't want to sacrifice any octopus after drawing enough blood for the study. And by chance, there were no female octopus were collected that met the mass requirement, so none were used in the study. And to understand the impact of ocean acidification and warming ocean temperature and the combination of these two factors, there are 4 immune system parameters in Octopus rubescens to examine. The 4 immune system parameters are: 1. The hemocyte count. Hemocyte is a blood cell, especially in invertebrates, which assists in wound repair, phagocytosis, and coagulation. And 2. The phagocytosis rate. The phagocytosis is an injection of bacteria or other material by phagocytes. 3. The lysozyme activity. Lysozyme is an enzyme that catalyzes the destruction of cell walls of certain bacteria. And 4. The superoxide production. And there were 4 target groups, and each octopus was randomly assigned to these 4 treatments. And 1 is control temperature, control carbon dioxide partial pressure. 2. The control temperature, the high carbon dioxide partial pressure. 3. The high temperature, the control partial pressure of carbon dioxide. And 4. High temperature, the high carbon dioxide partial pressure. And from the collection side, the partial pressure of carbon dioxide was 740 mu atm, and the temperature was 10.1 degrees Celsius, which become our control conditions. And the target treatment of carbon dioxide partial pressure was 530 mu atm higher than control, and the target treatment temperatures was 3.5 degrees Celsius higher than the control temperature treatment, both based on the RCP 8.5 prediction for the end of the century. And what is RCP? RCP representative concentration pathway. It's a greenhouse gas concentration trajectory adapted by the IPCC. And RCP 8.5 is generally taken as the basis of worst-case climate change scenario. And it is a likely outcome if the society does not make concerted effort to cut greenhouse gas emission. And here we come to the holding tank design. The tank system included a chiller and a venturi injector to maintain the proper temperature and oxygen partial pressure. Each aquarium had a slow, constant water exchange system, preventing buildup of ammonia or waste in each tank. And blood collection. Blood was collected 20 to 22 days after the designated treatment. Each octopus was anesthetized by submersion in 2.5% ethanol in seawater until the octopus was unresponsive. Blood was drawn from the branchial vein, and the target volume of blood was 0.5 milliliter. After blood extraction, a 20 milliliter aliquot of blood was used to perform a hemocyte count. The remainder of blood was centrifuged. Following centrifugation, the supernatant cell-free plasma was removed and stored on ice for the lysozyme visit. The isozyme in the past was washed with Artifice solution and concentrated. Lastly, the Octopus Ranger solution was removed and the hemocyte will be suspended in 1.0 milliliter modified tank balance solution and stored on ice for phagocytosis and superoxide production assays. And I have simplified a lot of procedures to the result. The study represents the first explanation of Cyberus immune response or immune response of any non-calcified mollusks to ocean acidification or warming. And after a 3-week exposure to elevated carbon dioxide partial pressure and or temperature, we found a significant increase in blood hemocyte count and total phagocytosis in response to elevated carbon dioxide. And different color indicates different temperature, and the left side shows a lower partial pressure of carbon dioxide, and the right side shows a higher partial pressure of carbon dioxide. Okay, and there's no difference in those parameters in response to temperature and difference in superoxide production or proportion of phagocytosis cells for either treatment or the combination of the treatments. And discussion. These results indicate that elevated carbon dioxide may cause a limited but important increase in immune response in octopus rubescens, while our research suggests that octopus rubescens may be fragile to ocean acidification. However, more studies on octopus physiology, especially longer term and throughout different life stages, are necessary to understand how this important ecological group will respond to future ocean conditions. Okay, and this is the end of my talk.
I want to see the slide of blood collection. And I want to know the word.
Thank you.
And the type. What this word means.
Branchial is, she wants to know what branchial mean. And in the
Sorry, again? Which word?
Mercury, right?
No. ANES.
Anesthetized. It is a procedure that usually in human we do before the operation. This is 麻醉 and anesthetize.
Can you say again why they don't use female octopus?
Oh, he wants to know why in this study there were no female octopus were used, because we have a mass requirement for the octopus, and just by chance there were no female octopus that meet the weight requirement. So that's why no female octopus were used in the experiment.
What's your name?
Can you explain what is the meaning of superoxide production?
Okay. Can you please repeat the question?
Can you explain what is the superoxide production?
Can I? He wants to know. He wants to get more detail about the superoxide production. Well, in the study they didn't mention too much about the superoxide production, so I don't know either. Neither do I. Okay.
Patrick.
Can you show me a chart in your... Yes. What is a pH? What is your Y scale in...
You mean
before this? You don't have only this chart, right?
I have two charts. This one and that.
Okay.
Which one?
Previous one.
Okay. This one?
Can you expand your chart again? Just please.
Okay, sure. The different colors indicate different temperature, and the right part shows a lower partial pressure of carbon dioxide, and the right shows a higher partial pressure of carbon dioxide. THC represents the total hemocyte count.
As you can see, that's another extreme. I think many of you start to smile when you see some slides like this. Okay, you know the problem is, right? Yeah, you also smile. Okay. So, you know, we don't want to read the paper. So you're asking us to read the paper. So, okay. What can you improve? Let's talk about some good things. So it's nice that you explained this RCP 8.5. I was actually thinking about what's that. Okay. And there was some different situation. For example, here you talk a lot about methods, and you print a lot. You see a lot of text. And here you talk about octopus, but you actually, and you said a lot, you talk a lot, but you didn't print keywords. So the level of words, the amounts of words should be comparable to your lines. So here you print too few keywords. That's the place where our minds drifted away. So please give us some keywords here. For example, you say a lot about the importance of octopus, but my mind was wandering away. So give us keywords. You have space here. All right. And then the word counts in total, I would say you should reduce by 50% and use bullets. You know what bullets is, right? Use bullets, and the method should be condensed and condensed and condensed. So you don't really need to explain how you withdraw blood from the octopus. That's not that important. Or how you do the centrifuge. That's also unimportant. What's important is you need to explain, that follow one of the questions here. You need to explain why you choose these 4 parameters. And I know it's difficult to explain this because these are medical terms. So how you can do that, what you can do is, first you talk about hemocytes, and then you show, I don't think this is hemocyte, where's the? You show a photo image of hemocytes, non-phagocytosis. Here, for hemocyte, you show a photo of hemocytes, and you explain if the hemocyte count is high in human body, what will happen, and if the hemocyte count is too low, what will happen. Okay, you know if the hemocyte count is low, you will feel dizzy from time to time, 贫血, okay? So, explain using the example of human bodies, and what's the importance of phagocytosis and superoxide. That was the question from the audience. You should give us an example. What is superoxide? And if the superoxide production is high, what does that mean? So, in my opinion, each of the parameters here should be 1 slide. Give us image and then explain its meaning in human body. So that's the place you need to add more. And the place you should reduce a lot is your methods. These are far too detailed, especially this. Okay. And if you really want to explain the experimental procedure, you can consult the talks from previous talk today. Use animations. You can draw a square and say this is the tank of the octopus. That would be very easy to do in PPT. And here, for the introduction of data, because you only have 5 data, in my opinion, each of the data should be, like the A, should be in 1 slide. Make it this big, and if possible, tell us again, what does please do not use abbreviation, just tell us this is total hemocyte counts. You see, I already forget. Tell us again if the count is high or low, what does that mean? And the same forward for all the 5 figures. Then you will easily make 10 minutes. Lastly, you end here. It's a little bit abrupt. Give us a conclusion. What was found in relation to the 4 parameters here? Which one is changed and which one not changed? Alright? Okay, so I think there's quite some space for improvement, and hope to see your improvement next time. Thank you, Alan.